Developing a bioprocess for a biological drug is very complex. The more you know about the process, the better you can respond when unexpected events occur. It is common to monitor critical process parameters like pH, dissolved oxygen, or viable cell density with inline sensors, and use a set of at-line analyzers for glucose, lactate, ammonia, etc. But it is less common to measure and monitor amino acid concentrations in the cell culture media, despite the wealth of knowledge and opportunities those measurements can offer. The lack of frequency might have a lot to do with conventional amino acid monitoring simply taking too long. The process involves pulling a sample, filtering out the cells, and then carefully securing, labeling, and freezing them for transport. Once the samples arrive at the core lab, they're defrosted, diluted, and prepped alongside derivatization reagents and chemicals. Once ready, they're loaded into the HPLC so that data can be acquired and analyzed, and this can take a long time. The results are then summarized in a report to be sent back to the bioprocess lab. This process can take as long as 3 to 6 weeks if the core lab is especially busy or dealing with a backlog. We often hear that by the time the results finally come back, the project is already done and the team has moved on, without the information they identified that they needed. This is why the Rebel at-line analyzer is a game changer for bioprocessing. It's faster, easier, and requires much lower sample volumes. With Rebel, you can pull a sample as small as 10 microliters, spin it and dilute, insert into the Rebel, name it and hit run. An automated report will be generated in as little as 10 minutes per sample. With the power to run this analysis at line, you can collect data much more often and use it to optimize your bioprocess faster. Curious if this will work in your lab?